A warm welcome to all of you. Today we are discussing Transpiration Part 2. Let us learn the kinds of transpiration. They are stomatal transpiration, cuticular transpiration, and lenticular transpiration. First one, stomatal transpiration. Before going to the mechanism of stomatal transpiration, we have to know the structure of stoma. This is a single stoma. Then what is stomata? More than one openings, we call it as stomata. Where it is located? Stomata are minute openings in the lower epidermal layer of leaves. A stoma is surrounded by a bean-shaped guard cells. Here you can see a single opening, we call it a stoma. Yes, more than one openings, together we call it a stomata. Then this is a stomatal apparatus, we can say. The structure of stoma or stomatal apparatus. Here you can see two guard cells are there. Two guard cells, that is in each shape is kidney shape or bean shaped guard cells you can see. Then that is present in dicot leaf. Then dicot leaf you can see kidney shaped guard cells or bean shaped guard cells. Where it is located? It is located in the lower epidermal layer of leaves. Here you can see this is the lower side of the leaf. Then lower epidermal layer of the leaves. What you can see? You can see the stoma or stomata. You can see in the lower epidermal layer of the leaves. Then this diagram is very important for examination, for drawing and labeling. Then you can label these parts. Guard cells. Yes, bean-shaped guard cells. Then thick inner wall, nucleus, chloroplast, thin outer wall. Then you can label epidermal cells. These two guard cells are surrounded by epidermal cells. Then you can label inner thick wall and outer thin wall. Then chloroplast, nucleus, vacuole. Then what is this? This is stomatal pore. This is open stoma. This is closed stoma. You can see here. Open stoma, closed stoma. Then these labelings are very important. You have to learn this. Is the more than one opening, we call it as stomata. That is located in the lower epidermal layer of the leaves. Very important. Then when these stomata will open during daytime, stomata are wide open primarily for the intake of carbon dioxide for photosynthesis. Daytime it will wide open. But there are some factors are there to open and close stomata that we will learn in the next slides. Then you can see number of stomata range from 1000 to 10,000 per square centimeter. These points you have to keep in mind. Mechanism of stomatal transpiration. In stomatal transpiration, water vapor escapes through the stomata of the leaves. Water after absorption by the roots from the soil rises up through the stem and reaches the tissues of the leaves through veins. You know that. A large number of spongy mesophyll cells you can see in the leaves. Here this is the cross section of leaf. Here you can see large number of spongy mesophyll cells. These cells we call it as palisade cells. Then these are next to palisade cell, that is that are spongy mesophyll cells. Here you can see is first layer is palisade cells, palisade mesophyll cells. Next to palisade mesophyll cells, you can see spongy mesophyll cells. They are loosely packed. You can see here. Then what will be there in this is spongy mesophyll cells, intercellular spaces you can see here. Then large number of spongy mesophyll cells in the leaves have their surface exposed to the intercellular spaces. Large number of intercellular spaces are there in between the spongy mesophyll cells because they are loosely packed. Here you can see that. These surfaces of the cell give out some of the water as a thin film. When is it possible? When the cells are turgid. The soil contains enough water, then only the cells become turgid. Then these surfaces of the cell give out some of the water as a thin film. The water from the film evaporates and water vapor diffuses into the intercellular spaces. Here you can see these are intercellular spaces. Then water vapor diffuses into the intercellular spaces. Finally, it reaches the substomatal space. These are the substomatal space from where it escapes through the stomata into the outside atmosphere by diffusion. This is the mechanism of stomatal transpiration. Here you can see that yes, this is substomatal space. Then this water vapor finally reaches the substomatal space. From the substomatal space, water vapor escape through the stomata and into the outside atmosphere by diffusion. The cells that lose water by transpiration replace it by drawing water from the nearest vein. Here you can see this is vein. This consists of xylem vessels. Most of this water travels along the cell walls, cell wall of one cell to the cell wall of another cell, like that. Then most of this water travels along the cell walls by imbibition, and the small quantity enters inside the cell by osmosis. Then two mechanisms are there to travel water along the cell walls by imbibition and enters the cell. Water enters the cell by osmosis. Thousands of leaf cells evaporate water by transpiration, causing more water to be pulled from below via xylem vessels. There are numerous number of stomata present on the undersurface of dicot leaf. You already learned that almost 1000 to 10,000 stomata present on the undersurface of dicot leaf per square centimeter. The transpiration pool can thus be created and can draw up water to about 50 meters or more in tall trees. More transpiration occurs on the undersurface of the dicot leaf. There are more stomatal openings on the undersurface of the dicot leaf. That's why more transpiration on the undersurface of the dicot leaf. Through experiments, we can see that. We can prove that yes, transpiration is more on the undersurface of the dicot leaf. Let us see that. More transpiration occurs from undersurface or lower surface of leaves. This can be proved by an experiment in which pieces of dry cobalt chloride paper are attached to the two surfaces of leaf and held in position by two glass slides on either side tied together by elastic bands. In this picture you can see clearly, yes, here this is upper surface, then this is lower surface of the leaf. Then both the surface we have to place cobalt chloride paper, dry cobalt chloride paper. Then this cobalt chloride paper held in position by two glass slides on either side and tied or held by clips. The leaf should remain attached to its own plant. That is very important. Yes, that is a very important point you have to remember while doing the experiment. The cobalt chloride paper should be dry, or that should be blue in color. Then the leaf should remain attached to its own plant. These 2 points you have to remember while doing the experiment. The pieces of paper which is facing the upper surface, here you can see upper surface of the leaf either does not turn pink or turns pink in much longer time. But in the case of cobalt chloride paper that placed on the lower surface of the leaf, this is the lower surface, then what happened to that cobalt chloride paper? That cobalt chloride paper turned pink. Why is it so? Because more stomata present on the, or numerous stomata present on the lower surface of the leaf, but here in the upper surface there is no stomata. But how is it turned pink after long time? Due to cuticular transpiration. Here on the lower surface, cuticular transpiration as well as stomatal transpiration is there. That's why that turned pink very fast, because more numerous stomata present on the lower surface. Then compared to stomatal transpiration, cuticular transpiration is very less. The amount of water vapor escaped through the cuticle, that is very less compared to stomata. Then the lower surface of the leaf, has more stomata. That's why cobalt chloride paper turned pink. Dry cobalt chloride paper on the lower surface of the dicot leaf turned pink. But dry cobalt chloride paper on the upper surface of the dicot leaf does not turn pink. It will take more time to turn pink, because here only cuticular transpiration on the upper surface, only cuticular transpiration takes place. That is the reason. Leaves of some plants wilt during midday and recover in the evening if there is plenty of water in the soil. Give the reason. Yes. For this statement, what will be the answer? During the midday, the rate of transpiration exceeds the rate of absorption of water by the roots. The rate of transpiration more than the rate of absorption of water by the roots. That's why leaves of some plants wilt during midday. Why is it recover in the evening? There is no answer for this statement. Yes. Then what will be the answer? That answer also you should include. In the evening, what will happen? In the evening, the rate of transpiration is less than the rate of absorption of water by the roots. That's why that will recover in the evening if there is plenty of water in the soil. Then these 2 points you have to Write in give reason question if the statement is like this. Stomatal regulation of transpiration. The opening and closing mechanism of stomata are regulated by the amount of water and solutes present in the guard cells. Here, yes, you can see the open stoma, then closed stoma. This is closed stoma. Then the opening and closing mechanism of stomata regulated by the amount of water and solutes present in the guard cells. You can see here, these are guard cells. Then another point, transpiration occurs as long as the stomata are open but stops when they are closed. Stomatal transpiration is regulated by adjusting the size of the stoma. The next type of transpiration is cuticular transpiration. What is cuticle? The cuticle is a waxy layer secreted by the epidermis on the two surfaces of the leaf. Here you can see this is the section of leaf, upper and lower epidermis you can see here. Then a waxy layer secreted by the epidermis. This waxy layer is next to the epidermis, above the upper epidermis and below the lower epidermis. You can see cuticle, that is a waxy layer. Then what is the function of this waxy layer or cuticle? To prevent evaporation from the leaf surfaces of the leaf. That is the function of cuticle. Then greater the thickness of the cuticle, lesser the transpiration. Then based on the thickness of the cuticle, the transpiration rate will change. Then in desert plant, example, in desert plants have thicker cuticle to cut down transpiration. Then you can see here, this is cuticular layer. This is secreted by the epidermis on the two surfaces of the leaf. Here you can see, yes, this is the lower epidermis, this is upper epidermis. Then both the surfaces of the leaf, a waxy coating is there. What is the function of this waxy coating? To prevent evaporation from the leaf surfaces of the leaf. The next type of transpiration is lenticular transpiration. Before going to the lenticular transpiration, you have to remember the term lenticels. Lenticels are special openings that develop on the bark of older stems. These lenticels are special openings. Here you can see these openings, we call it as lenticels. Spellings also you have to remember is where is it located? Where is the location of lenticels? On the bark of older stems. Here you can see the lenticel. Yes. Then instead of stomata, what is there in the older stems? Lenticels are there. These are special openings. Develop on the bark of older stems. What are the functions of these lenticels? This will allow diffusion of gases for respiration as well as photosynthesis. Because these lenticels never close. They remain open all the time. Then this will allow diffusion of gases for respiration as well as photosynthesis. Lenticels never close. Very important point. They remain open all the time. Then what is lenticular transpiration? Water from the cell surface directly facing the lenticel. Here you can see water from the cell surface directly facing the lenticel evaporates and contributes to transpiration. This process is called lenticular transpiration. Comparison of types of transpiration based on the amount of water vapor. There are three types of transpiration are there based on the amount of water vapor escaped from the plants to the atmosphere. They are stomatal transpiration, then lenticular transpiration and cuticular transpiration. If you are comparing these three transpiration, which one is more? Stomatal transpiration is greater than lenticular transpiration. Lenticular transpiration is greater than cuticular transpiration. This order is very important for examination. You have to remember this. Can you identify these leaves? These leaves are dicot leaves because these showing reticulate venation. If you observe these leaves, you can see the upper surface and lower surface of the leaves. In the lower surface, here these two pictures showing the lower surface, here veins are very clear. More stomata are present on the lower surface of the leaf. Then upper surface, there is no stomata, but cuticular transpiration takes place in the upper surface. But in the lower surface, stomatal transpiration as well as cuticular transpiration takes place. Then in which part more transpiration takes place? Of course, that is in the lower surface of the leaf. Revision. Answer the following. First question. From the following list, pick out the parts through which the water vapor of transpiration leaves the leaf and rearrange them in proper sequence. Here they are giving some parts of the leaf: xylem vessels, mesophyll cells, stoma, intercellular space and substomatal space. Based on the clue, you have to arrange these parts in proper sequence. Here they are given water vapor of transpiration leaves the leaf. That is the clue. Then based on that, you have to arrange these parts in proper sequence. Then how to arrange that? Xylem vessels, then mesophyll cells, intercellular space, substomatal space and stoma. In this way, you can arrange the parts. Answer: xylem vessels, mesophyll cells, intercellular space, substomatal space and stoma. Thank you, children. Have a good day. If you like my channel, please like, share and subscribe. Thank you.